So we left off last time with an overview of DNA replication. And I'm just going to go over that real quickly for you again, because I'm sure it's probably a little bit confusing still at this point. So here on the left, we have our DNA replication fork. And our double-stranded DNA is up at the top here with our fork down here. And we have our topoisomerases leading the pack, leading the way for the helicases to kind of uncoil the supercoiling of the double-stranded DNA. Our helicases come in and they separate the two strands of the DNA. And in doing so, they make single-stranded DNA available, which the single-stranded binding proteins, or SBB, binds to not allow for any secondary structure or reannealing of the single-stranded DNA. We also have our leading strand, which is synthesized continuously with our leading strand DNA polymerase. And on our lagging strand, we have discontinuous synthesis, which our RNA primase lays down primers made of RNA, and then our lagging strand DNA polymerase synthesizes DNA in a discontinuous manner. So in doing so, when we remove the RNA primer, we have little nicks that we need to ligate together with DNA ligase. So we went over a lot of the DNA replication proteins, and we talked about DNA polymerase and the different problems that it faces when trying to synthesize DNA. Now we're going to talk about a different problem called the end replication problem. And this comes about only in eukaryotic cells just because we have linear DNA. So this does not occur in cellular DNA because they do not have the ends on their DNA. So in this figure, we have the parental DNA, or the old template strand, in blue, and our purple is our newly synthesized DNA. Now, in this, going from the 5' to 3' end is fine. This is our continuous leading strand synthesis. And this is great because we can go all the way to the very end, replicate up through the 5' end of the template strand. And this is done, again, in the 5' to 3' direction. And everything is fine and dandy with this type of, with the leading strand. However, when we get, that was the lagging strand. However, when we get to the 3' end of the template, or the 5' end of the newly synthesized DNA, we have a problem. So as we remember, we have our RNA primer. What happens is that this primer is removed by an RNase. But if you remember, DNA polymerase cannot initiate synthesis on its own. It needs a 3' hydroxide group in order to do this. And again, it cannot synthesize in the 3' to 5' direction. So what we have is when this RNase removes the primer, there's nothing left, and we cannot synthesize over that gap. So we're left with a single-stranded DNA overhang. And this can be very detrimental to our cells. And this is because in our cells, we have nucleases. And what nucleases do is they chew up or cleave single-stranded DNA. And these are very nonspecific. They will cleave any sort of single-stranded DNA that's around. And so since we have our 3' overhangs, our nuclease will come in and cut that out. And we will come up with very short DNA on both sides. And in both copies, we will have shortened DNA. So for each round of replication, we're going to be shortening our chromosomes. And as you can imagine, this can be very detrimental to the cells because we can be cutting into very important genes that we need in order to survive. So what we have are called telomeres that prevent the chewing up of our chromosomes. So telomeres are extensions of chromosomes. What they are the repeating sequences of DNA that are added at the 3' end at the end of replication. These sequences are TT, A, GGG sequences, and they do not encode for anything. They are not necessary for our cells to survive. So we can just add as many of these on to the 3' end of our template strand as needed. And they're dispensable, so we can remove them and add as many as we need to. So what happens is what's called a telomerase an enzyme can come in and actually add on these repeating subunits, or sorry, these repeating units of DNA. And then our DNA polymerase can come in and in the 5' to 3' direction fill in this and make double-stranded DNA. Now we're still left over with the 3' single-stranded DNA on our template strand, but this is okay because, as I said, telomeres are dispensable. They do not code for any proteins, so this is okay. This is not a problem. And these telomeres can be very long in our cells, anywhere from 5 to 15,000 base pairs long. And interestingly, as infants, we have more towards the 15,000 base pair mark and closer to 5 as the older we get. And this is because the enzyme that that increases our telomere length actually decreases with age. And we'll get into that a little bit later. So as I said, telomerase is the enzyme that adds telomeres to the 3' end of our DNA after replication. And what telomerase is a ribonucleoprotein. And as the name suggests, it has a ribonucleo component as well as a protein component. The protein component is called a reverse transcriptase. And as most of you know, in transcription, we use a DNA template, and we make RNA from that. That's called transcription. However, as you can imagine, in reverse transcriptase, we take RNA and use that as a template and make DNA. So it's very interesting is our telomerase has an RNA component which uses its own, it actually contains an RNA primer in this part of the enzyme. So it uses its own RNA primer to transcribe DNA. So it'll lay down and over it'll add these repeats. So then after replication, we can just take these 3' ends off, and we will not have a problem. And this protects the genes within our chromosomes. So once telomeres were identified, people were really excited. They thought, hey, we found the key to immortality or the fountain of youth. Well, this isn't exactly the case, but as you'll see in a little bit. But so we are composed of mortal cells, which means that telomerase can be inactivated. So as I said, the older we get, the less active our telomerase can become. Thus, if we have inactive telomerase, telomeres, the telomere length will decrease with each round of replication. So what can happen is that after we keep taking off ends of our chromosomes after every round of replication, we're going to be cutting into our genes. And when this happens, cells will reach what's called a senescence stage, in which the cells will no longer be able to divide, and essentially the cells will die. Thus, they're mortal. They're also what's called immortal cells, and this is where these cells contain active telomerase. Cancer cells often have very active telomerase. So since they have active telomerase, their DNA ends will be maintained throughout DNA replication, no matter how many rounds they go through, and thus the chromosomes will be maintained as well. And so the cells continue to live. Now, interestingly, there's a bird, I think it's called the leech storm petrel, which, interestingly, it has very long telomeres. And it was identified that this bird actually lives a lot longer lifetime than normal birds. So they thought, oh, we found the key, the key to immortality. All we got to do is just activate telomerase, and we're going to live forever. Well, that's not exactly what we can do, because there's a lot of different things that go into aging. And we have cancer can occur. We have DNA mutations that can lead to cancer. We can, apoptosis can happen. So there are a lot of different things that'll happen in order for our cells to die. Now, one protein or enzyme that's very similar to telomerase is HIV reverse transcriptase. This is a bifunctional protein. Here's the structure. And it contains both a polymerase and an RNase component. So the polymerase has the same structure as I explained before, where it has kind of like a right hand, a palm, thumb, and fingers domain. It also has an RNase component, which can cleave RNA. So what it does is it can use either DNA or RNA as its template to make more DNA. So let's say it can use RNA as a template and produce DNA from that, so it'll make a DNA-RNA hybrid. What happens is then the RNase component comes in and cleaves the RNA template, resulting in single-stranded DNA. And after that, the polymerase, again, can come in and do a second round of synthesis to make a complement to that DNA, resulting in now double-stranded DNA. So we went from single-stranded RNA to double-stranded DNA, all with this one enzyme. And it's thought that this could have derived from telomerase. And we're going to switch gears a little bit for the rest of the chapter and talk about DNA packaging. DNA packaging affects a lot of different things in the cell, such as activity of gene expression. So here's a picture of a eukaryotic cell, or sorry, eukaryotic nucleus, and it's been stained for different types of DNA. In the red is heterochromatin, and in the yellow is euchromatin. Sorry, these arrows are a little off. But anyway, heterochromatin is it's called heterochromatin because it's tightly packed DNA. It's very compacted. Because it's very compacted, it cannot allow for transcription factors to come in and bind, and so it's not transcribed. The genes are not transcribed. And when genes are not transcribed, it's also known as silencing or repressing the genes. And it's found that it was identified that heterochromatin is most often found on the periphery of the nucleus or around the outside edges of the nucleus. So it seems like there's some correlation between the packaging of the DNA, the transcription or activity of the DNA, as well as the location. So in euchromatin, this is a looser DNA, so it's less compact, so that RNA transcription machinery can come in and bind to the DNA, thus allowing transcription and translation to occur. And this was actually found near the center of the nucleus. So again, there seems to be a correlation between the packaging of the DNA, transcription activity, whether it's actively transcribed or silenced, or in the location of the DNA as well. Now let's get into compacting a little further. This is the nucleosome is the main repeating structural subunit of eukaryotic chromosomes. One nucleosome is composed of DNA and proteins. So here's the structure. We have our DNA is helix, and it's this red helix. And the proteins called histones is what the DNA wraps around. And there are 8 histone proteins, and it makes up an octamer. So what nucleosomes do is they compact the DNA. And we actually need to compact it even further than this, because our DNA is 1 meter in length, which is huge compared to our nucleus, which is between 5 and 10 microns long. So if you can imagine, our full-length DNA is 1 million times the size of the nucleus. So we need to compact. It's almost unimaginable how compact our DNA needs to get. Now, the compaction of the DNA also regulates transcription. And by doing so, again, I said, the more access that we have to the DNA, the more likely it is to be transcribed. So as you can probably imagine, the DNA that's wrapped around the histone proteins are less accessible to protein binding than the DNA that's just freely hanging around. Nucleosomes are also known as, can be called beads on a string model. That's because I'm sure it's easy to see that it's kind of like beads on a string. So the nucleosome core consists of, again, the 8 histone proteins and the DNA. So the 8 histone proteins are 2 copies each of H2A, H2B, H3, and H4. So there are 2 copies of H2A, 2 of H2B, and so on. Now H2A and H2B actually, they pair together, as well as H3 and H4 pairs together. And then these pairs come together in the center of the nucleosome. And the DNA wraps around the histone in approximately two times. As shown here, we have our DNA in blue, and then the nucleosome core histone proteins in the center. And here's another way to look at it. We have a crystal structure of the DNA in dark blue, and then the histones on the inside. And it's not visible with this model, but histones have very long N-terminal tails that stick out from the nucleosome core. And this can be seen here, as well as here in this figure. And these can be very long. And the reason that we don't see them in this figure is because they're very flexible. And when we do X-ray crystallography, we need something to be very stable. And it's very hard to see anything that's flexible in crystallography. But trust me, they're there. Anyway, these tails interact with DNA. And in doing so, they can make the DNA more compact, as we will see later on. Are there any questions before I go any further? All right, so here's another view of the nucleosome. We have our DNA wraps around the nucleus. Hopefully this is visible on your handouts. We also have our histones. Yellow is H2A, and red is H2B, blue is H3, and green is H4. And like I said, the H2A and H2B fold around or pair up with each other, as well as the H3 and H4. And again, you can see these tails, and these aren't the full-length tails, but this is part of the tails. They are jutting out from the center. Let's see, here we got another one over here. And they are interacting with the DNA to help compact the DNA. So the complete nucleosome adds another linker histone. So as I talked about up to now is that there are 146 base pairs of DNA wrapped around the nucleosome, which consists of 8 histone core proteins. There's one more histone protein called the linker histone, which is called H1. And this binds DNA between the nucleosomes. And so we have our linker DNA here between each of the nucleosomes. And the histone binds that and stabilizes the compaction even further. And then it compacts the nucleosomes even further. So overall, nucleosomes compact the DNA by 30 to 40 times. So what that means is that if we had our DNA stretched out, it would be 30 to 40 times larger than when we had these histone proteins around to compact it. So even further compaction happens allows these to fold into even smaller or more compact structures called solenoids. So we want DNA to be compacted prior to cell division, because we don't want our DNA to be flopping around, tangled up with itself or other proteins or anything else, or get damaged in any way or cleaved. So prior to cell division, we compact our DNA. But what happens is that nucleosomes fold around each other and form irregular shaped solenoids. And these are extremely short rods. They're about 10 micrometers long. And again, it prevents the tangling of the DNA and prevents anything else from happening to the DNA that we don't want to happen. And since they're so small, they're easily moved around the cell. As I'm sure we've all seen with during mitosis, we want these nucleosomes to be moved easily. However, since it's so compact, it's not available for replication or transcription until it's unpacked. But this is good because during mitosis, we don't want, when cells are dividing, we don't want anything to be transcribed or translated into proteins. As it turns out, solenoids are visible using electron microscopy, as seen over here. And they actually do look like beads on a string. And these can actually, the amount of compaction can change depending on the environment that the solenoids are in. So here we have a more compact solenoid. They can also be a little bit less compact, such as in this figure or this figure up here. So different variables can change the degree of compaction. And this includes salt, the amount of salt that's in the environment, the amount of linker histone that's around that can bind to the DNA between the nucleosomes, as well as many different histone modifications that can occur, which we'll talk about in more detail in a little bit, as well as the binding of other proteins or the supercoiling of the DNA. When DNA is in the solenoid form, it follows a very convoluted path in the solenoid. It's not very regular. It's very hard to follow the path. And something more importantly is that this compaction also affects what surfaces of histones and DNA are accessible by other proteins. So as you can imagine, these red strings are the DNA. So as you can imagine, this DNA is a lot more accessible to proteins or other transcription machinery, a lot more so than the DNA that's within the center or in between nucleosomes, as well as the surfaces of the histones, such as the DNA tails. So you can imagine the tails that are sticking out in this side of the nucleosome are going to be a lot more accessible than those within the center. So let's get into even higher orders of packing. We start with our double-stranded DNA, which is in a helix, which is a little bit compacted, and then we put that into more compaction, beads on the string, or nucleosomes. which are looped around each other even further to make 30 nanometer solenoids, which again loop around each other further and make little sections of chromosomes, which again, these sections will loop around each other and coil even further to be sections of chromosomes. And this big piece right here is just this little part of the entire chromosome. So overall, each DNA molecule has been packaged into a chromosome that is 50,000 times shorter than its normal length as when it's linear. Now, I don't know about you guys, I think that's amazing. So there are a lot of different things that can alter the structure of chromosomes. And there are three mechanisms that alter chromatin, and these are histone modification, which we'll be spending a lot of time talking about, histone remodeling complexes, as well as the replacement of core histones with histone variants. First, let's talk about histone modifications. As I said before, histone tails are very flexible and protrude from the surface of a nucleosome. And these can be very long and they have a lot of residues that may be affected. And these tails can be modified in many ways on specific residues. So in one nucleosome, we have 8 core histone proteins. So we have so that means we have 8 tails that are able to be modified. So we have 2 H2A tails, 2 H2B tails, H3 and H4. So for example, on H2A tail, we have, there's a serine at residue 1 that can be modified. We have lysines at residues 5 and 9 that can be modified. Anyway, just to get you the idea, there are so many different modifications that can occur on a lot of different residues on these proteins. So different modifications that can occur are acetylation and methylation, which are the two that we'll mainly be focusing on today, ubiquitination, sumoylation, and phosphorylation. And here's a figure of 2 histone proteins interacting. So for instance, it could be H2A and H2B. So what they do is they have a histone fold, which consists of an alpha helix, loop, alpha helix, loop, alpha helix. And you can't see the tails in this figure, but the tails is what is being modified. And these are modified covalently. So on lysines, we can have acetylation or methylation. And on serines and threonines, we can have phosphorylation. And these marks mean this is acetylation, here's methylation. And it has been hypothesized that there exists a histone code. And what is meant by this is that the code of these different marks, these different modifications, are recognized by different proteins. And they affect the protein-DNA interactions to allow for transcription or the lack thereof. So, for instance, acetylation is identified by the bromo domain of certain proteins. Methylation is identified by the chromo domain of different proteins. However, it's a very controversial thing in the field right now because we don't know if there actually is a code. Many people think that that there is a specific code that whenever you acetylate something, it's openly easy to be transcribed. However, there's a lot of instances where this isn't true. So it's a very controversial thing right now. So let's look at acetylation. Again, modification alters chromatin structure, and it also, by doing so, affects gene expression. So let's look into that a little bit further. Histone acetyltransferases, or HATs, are the enzymes that add acetyl groups to our histone tails. So here we have a nucleosome. The blue represents the DNA, and the green represents the histone tails. So the histone tails are, in this figure, they're interacting with the DNA. Now, when we add our acetyl groups from the HATs, these histone tails can be removed from the center of the nucleosome. And you can imagine this is the case because lysine is positively charged at physiological pH. But when we add a acetyl group, it removes a positive charge and adds a neutral charge. So maybe it's thought that maybe the histone tails are being repulsed by the negative charge of the DNA. So whenever these histone tails are acetylated, it's thought that it usually opens the chromatin so that the RNA transcription machinery can come in and transcribe the DNA. However, this is not always the case. There are a lot of different instances where acetylation actually results in silencing of genes. So I would say more often than not, acetylation results in transcription, but not always. Just for the sake of this class, though, you just know that usually the acetylation results in active transcription of genes. There also exist histone deacetylases, which remove acetyl groups from histone tails. And what this usually does is it takes off the acetyl groups, thus compacting the nucleosomes again, so that the tails interact with the DNA, resulting in silent genes. So let's look at that a little further. Over here we have our acetylated histone tails, which result in an open form of the chromatin so that they can be actively transcribed. Over to the left we have unacetylated histone tails, so that they are more compact and results in gene silencing. Another modification that can occur is methylation. And methylation is put on histone tails by histone methyltransferases, or HMTs. And lysines can be either mono, di, or trimethylated. And here's an example of a trimethylated lysine. Here, note that there still is that positive charge on the lysine, so that they can still interact with the DNA tails. So most of the time, when we think of methylation, we think of gene silencing. Now, remember that sometimes this is not the case, that sometimes methylation results in actively transcribing genes, but for this class, just remember that methylation usually results in silencing of genes. And it's also recently identified. We didn't think that methylation could be removed from histone tails. However, it's been recently identified that demethylases or demethyltransferases actually exist. And also one thing to point out is that a single lysine residue can be modified with different chemical groups, such that we can have one lysine that an acetylation, or a HAT can come in and acetylate it, and then HDAC can come in and take the acetyl group off, and then histone methyltransferase can come in and put methylation on that same exact residue. Just something to keep in mind. So I said there are 3 different ways to alter chromatin structure. And we just talked about histone modification. I'm going to go a little bit into the other 2, histone remodeling and histone variants, or the replacement of histone core proteins with histone variants. Oops. So I just talked about modification. Here's our nucleosome. We have our DNA wrapped around, and here are our histone tails that can be modified covalently. And histone remodelers, what they do is they actually change the position of the DNA around histones. So in this case, we have this regulatory protein that wants to bind to this section of the DNA. But since it's tightly packed in the nucleosome, it cannot access that part of the DNA. So with the use of histone remodelers, the DNA can actually be moved around the nucleosome so that these regulatory proteins can bind at that specific site of the DNA. Another thing that can happen is there are histone variants that exist that can replace core histones during different processes within the cells. So here we have our normal histone, and then some of the histones have been replaced with their variants. One example in humans is H2A is replaced by H2AX during specific times of DNA repair. Now, modification can also occur on DNA. And again, we have methylation. And methylation usually occurs on carbon 5 of cytosine. And it's interesting because this actually is normally thought to be associated with silencing of genes again, silencing transcription. So what happens is this methyl group adds to the carbon 5 of the cytosine and actually sticks into the major group of the DNA. And which you can imagine will alter DNA-protein interactions because so the proteins that interact with the major group of the DNA will not be able to bind in the correct way anymore, which would usually result in the silencing of these genes so that transcription cannot occur. Now, sequences called CpG islands, C stands for cytosine, G stands for guanine, and the P is just the phosphodiester backbone that occurs between the nucleosomes, or sorry, the nucleotides. Anyway, these sequences are often targeted for methylation. And near the start of most genes, we have what are called CpG islands. And if these CpG islands are unmethylated, they're usually actively transcribed. However, if they are methylated, that usually results in silencing. So again, methylation on histones as well as on DNA can be a marker for silent DNA. So we got to think about, we have methylation already on our parental copies of our DNA, and we want to continue that specific pattern onto our both copies of our DNA. So we have a little problem because we have our double-strand DNA, and it can be methylated on both strands. But we need to separate these strands in order to replicate our DNA. So that one copy will get one type of methylation, and the other copy will get the other methylation in this situation. So what happens is that we have maintenance methylases, which are enzymes that actually that pass on the methylation groups, so that our silencing traits as well as our transcriptionally active traits are passed on from parent to daughter cells. And this can be seen in genomic imprinting, which many of you may have heard about, in which certain genes within our cells, I think there are about 80 in humans and mice, which these alleles we have alleles passed on from our mother and our father, and specific alleles, specific genes have only one allele active. So let's say that in one of these genes, only our allele from our mother is actively transcribed, while the one from our father is silenced, and vice versa for other type of genes. And so this way, and if we have both, there are cases in which we have both of these alleles activated. This can be a very bad thing. We can get a lot of different placental developmental problems, things like that. So it's one thing I think is kind of cool is that this is involved in when we make hybrids. So we take two organisms and we breed them, and we get a hybrid. And a lot of times they'll have different kinds of expression patterns because they'll have methylation patterns from one organism and from another, which can together be something different than we've ever seen before. Now, sometimes modification is not a good thing, such as in our DNA. Again, we have our 5-methylcytosine, which is methylated on our carbon 5. Now, sometimes what can happen is naturally, cytosine can be deaminated spontaneously, in which case that this amine group actually becomes an oxygen group. And when this happens, you may notice that this new residue is actually a thymine, or this new nucleotide. So with the deamination, we actually get a point mutation from a CG base pair to a TA base pair, which can result in mutation and tumorigenesis potentially. So let's go back to chromosomes for a little bit. During interphase, chromosomes are not usually visible with electron microscopy, and that's because they are not condensed. They're being actively transcribed, so we cannot visualize them under the microscope. However, one interesting fact is that females, as we all know, in mammals have 2 X chromosomes. However, as many of you might not know, is we only need one of these chromosomes. So the other one is always inactivated, and this inactivation occurs randomly. It's not really chosen one way or another. And usually when one X is silenced in a cell, it's silenced for that entire lifespan of the cell. And because it's inactivated, again, it's compacted into a more dense structure, so it's actually visible during interphase, as seen in this figure here. So these dark bodies are the inactivated X, and these are called bar bodies. So the compaction can actually happen due to hypoacetylation, which is the downregulation or the lack of cell groups, or when CpGs are methylated. Another way it can occur is through exist RNA, and this is a very complicated process, but for the sake of the class, just know that what this does is it kind of coats the DNA so that transcription factors cannot bind and it cannot be transcribed. There are a couple diseases that result in, and it makes it very difficult to define whether somebody is male or female. So for instance, in Klinefelter's disease, this occurs in a male in which they have 2 X chromosomes and 1 Y chromosome. This happens in every 1 in 500 male births. And since they have 2 X chromosomes, one of them needs to be silenced. And so these males have a bar body so that they result in 1 active X gene and 1 inactive. There's another, and this results in reduced fertility in men as well. Turner's results when a female has only 1 X chromosome, and they just never got their other X chromosome. So they do not have a bar body because they have 1 chromosome and 1 X chromosome, and they don't need to silence another one. And this usually results in 1 in every 2,500 female births And there doesn't seem to be a lot of problems with these females, except for their sexual characteristics are a little bit underdeveloped. Now, it can exist where a male has two X chromosomes. And you may think, wait, there's two X chromosomes, this is just a female. You know, there's nothing wrong. However, what happened is during mitosis, or meiosis, sorry, in the Y chromosome, there's a gene called the SRY gene, which results in a lot of male characteristics. So what happened is that this SRY gene was actually translocated into an X chromosome. So this male still is a male because it still has that gene, which gives it a lot of its male characteristics. And since it has one X gene or one X chromosome and one that kind of resembles a Y chromosome, there's no bar body in these males. And one thing I think is kind of cool, I don't know if you guys know what a tortoiseshell cat is. But anyway, in females, you know, they have different patches on their body that are either black or orange. So they have two X chromosomes in females like we do. And in one of their chromosomes, there is an allele that codes for the orange color, and the other one codes for the black color. So if you take a specific patch of their fur, you can tell which X chromosome is actually being inactivated at the time. So if they have a patch of yellow or of orange color, we know that the X chromosome, which can contain the allele for the black gene, has been silenced. So one thing that's been identified pretty recently is we were able to actually sort chromosomes using flow cytometry. And what flow cytometry is it's a way to analyze different fluorescence probes that attach to specific sequences. In other words, something similar to antibodies. So what this does is we can actually karyotype our chromosomes. And by that I mean that we can, so we have our different colors for each different chromosome, and we can align these based on their size and the positioning of the centromere. And in doing so, we can actually label each chromosome with a different color, otherwise known as chromosome painting. And if you look down here, you can see that you know, there's a little orange on this blue chromosome, and there's a little bit of the chromosome 6 put onto chromosome 8. This results from homologous recombination during meiosis, which results in a lot of the diversity in our cultures. So due to this chromosome painting, what we get is this ability has led us to look at where these chromosomes exist in our nucleus, in our nuclei. So for instance, this pink chromosome, we can see it's exactly where it's located. So this has led us to do a lot of different experiments and led us to see which chromosomes contain more actively transcribed genes and which ones are maybe more silenced. So last I wanted to talk to you a little bit about the C-value paradox. And this doesn't have a lot to do with chromosomes, but it's an interesting thing nonetheless. And this comes about because the total amount of DNA in an organism doesn't always match up with the complexity of that organism. So for example, here's a figure. We have the size, the number of base pairs in the genome going from smallest on the left to more base pairs on the right. We have E. coli down here with about 4.2 megabase pairs, Drosophila 165 megabase pairs, and then Homo sapiens up here in the red with 1,600 megabase pairs. And you can see flowering plants actually are able to have more DNA content than we do, even though, as many of you probably would hope, we are more complex than flowering plants. And it's also seen that similar organisms can vary in their DNA content by 100 times, which is very perplexing. We don't really know what's going on here. We don't know if it reflects some sort of function within the organisms. And we know that a lot of our DNA isn't being transcribed, and we don't know if this is affected or not. And also, it could also be an accident that occurred throughout history. So don't pack up yet. I still have 10 minutes for you guys. This won't take 10 minutes, but we can slow it down a little bit. This is a video showing just how compact DNA really is inside our cells. And I don't know if I'm going to get any volume here, but I think this is a really cool video. It really shows you the amount of compaction that actually occurs. So here we have our DNA. Guess we're not getting any volume. Here's our double helix of DNA, and our histone proteins are coming in soon. There they are. So this is another computer simulation. Obviously, we couldn't take this from a real cell. Nucleosome, these on the strings are forming. And now these are kind of looping together in solenoids. Short. And already pretty compacted if you think about it. What happens is you loop even more around each. Kind of like a telephone cord. So this level of compaction is very tight. And then that even loops itself again. And then, which can be during this is the I showed you on Monday. It will be put up on Icon, so you can